All right, people, I'm going to jump right in. So I don't have a pre-slide to this one. We're just going straight for notes and lecture. So we've covered simple diffusion in the previous lecture. You should keep in mind that osmosis is the diffusion of water from an area with a low concentration of solute to an area with a high solute concentration. There are instances where organisms need to move solutes into or out of the cell with the help of a channel protein. So when this is the case, we describe that type of diffusion as facilitated diffusion. So if you think about the definition of facilitation or a facilitator, there's someone who makes something easier. And one way to remember the term is to just keep in mind that definition. So the word facilitate actually has some Latin origin. Facilis means easy. And then if you've ever taken a Spanish class, or if you're like a native Spanish speaker, you may have heard something being referred to or described as facile, like it just means easy. So these channel proteins, they help with the transport. They make it easier. And two channel proteins that we encounter when we look at membrane-bound proteins are aquaporins. And for this one, you should think about aqua is like water. And then that's their job, to help move water into or out of a cell. And you have another channel protein that we see mentioned, and that is an ion channel protein. So what they do is they open and they close when they're exposed to like specific stimuli. So notice that they're also referred to here as gated channels. Alright, this video is one that we saw in the previous lecture for this chapter. I believe it was the first lecture. And you can, when I hit play, actually I'll hit play and I'll talk at the same time. So what we see is the phospholipid bilayer. So you see those green stringy looking tentacles. That's the fatty acid portion of the phospholipid. And then you have the larger green molecules, which are the phosphate groups that they're attached to. And then in the center, you can see these blue helical shaped aquaporins. And what you should notice is that there is a bunch of water that's bustling on both sides of the membrane. And if you watch closely, you can see the water molecules shuttle through. So yeah, there you go, aquaporin. Alright, when we look at the mechanism by which the carrier proteins move ions into and out of a cell, we note that there's a slight change in the protein. And hi, say hi.
Yeah.
This is my husband. His name is Speedy. Okay, I'm talking again now about this. So anyway, alright, basically what we see is that with the carrier proteins, they're moving these ions into and out of a cell. And what we see is that there's a slight change in the protein that actually helps to push the solute that binds to that carrier protein from the outside of the cell in, or vice versa, so from the inside of the cell out. The shape change doesn't really require any energy. Actually, it requires no energy, and the molecule is just able to trigger the protein shape to change by binding with it, and so it pushes the solute straight through to wherever it needs to go. Alright, so we're looking at figure 5.13. There are two types of transport proteins that carry out facilitated diffusion on this image. So when we look at figure 8, notice that the channel appears to just stay open, but also notice that the solute is only moving one way, and that's into the cell. And then if we look at figure B, so in figure B, the carrier proteins, they may be open on the outward facing side of the membrane, but when the solute comes and binds to it, my cousin is so loud, it's ugly. Okay. And when the solute binds to it, what happens is the protein, it opens up, or rather it closes up on the once open side, and then it opens up inward. So it's kind of like if you were to put your hands together and then do it so that you're making like a V with your hands. And if you were to then close your hands, so put them right on top of each other and open the end part where your wrists are. It's easier for me to show you this in class. So I will do that. Thank you. Food. Alright, so sometimes what you're going to see is that organisms need to be able to move solutes against the concentration gradient. And all this means is that diffusion is not running its natural course. So when this happens, passive transport is not something that we are messing with. Instead, there's another type of diffusion, and it's called active transport, and we have to utilize it. Alright, so when we're talking about active transport, we're talking about a type of transport that requires energy. So since you are moving molecules against the natural flow that they would have, it's like you are forcing something to happen. So one way to think about this, and I'm really not trying to be funny, but like if you think about a relationship, and it doesn't have to be a romantic relationship, of course, it could be a friendship, it could even be like a parent. But anyway, you have this relationship and maybe you think that the relationship is not really working. You know, lack of communication, it could be a number of things that's affecting said relationship, but basically you and the other individual are not necessarily happy with how things are, and you might not really want to be stuck together. But let's say that you can't completely push that person out of your life. So what you would have to do is force yourself to continue on. And this is how active transport works. So you have these molecules that are in close proximity to each other. They really don't want to be. However, if there's nowhere else for them to go, they have no choice but to be stuck in the same environment. And so what the active transport mechanism does is push even more of the solute into that space that's already heavily occupied by solute. Alright, so you might be thinking, well, why would you want to force variations in concentration? Like, why not just let things diffuse naturally? This is what you need to remember. Not all cells are like those red blood cells that we saw in the osmosis lecture. So they don't all necessarily thrive in an isotonic environment, meaning like, you know, equal concentrations of water and solute on both sides, in the cell and out the cell. So not all cells thrive in that environment. One example would be like neurons. So when a neuron fires, it's depending on the buildup of like certain concentration gradients in order for the spark, if you will, to happen. So solutes, they need to reach a certain level to make things happen. And the most well-known active transport situation where we have to do like a buildup of one thing to get a result is called the sodium-potassium pump. And it literally involves the two elements, sodium and potassium. So that's why it's called that. It fits. So on the next slide, you're going to get an explanation of how this particular pump works.
Sometimes a cell needs to move a solute against its concentration gradient. This process is called active transport, and it requires input of energy from ATP. For instance, most animal cells need to expel sodium ions, Na+, and take in potassium ions, K+, both against their concentration gradients. Here is how the sodium-potassium pump works. Sodium ions bind to a transport protein. ATP transfers a phosphate group to the protein, providing the energy that causes the protein to change shape and push the sodium ions across the membrane, where they are released outside the cell. Potassium ions now bind to the transport protein, and the phosphate group is released. This causes the protein to return to its original shape, releasing the potassium ions inside the cell. The transport protein is now ready to repeat the process.
Okay, guys, you know we gotta love her. Bioflix lady, you're awesome. Alright, so in this slide, we can watch a chemical, the process happening chemically with this 3D model. So check this out. So we're at the membrane. We're looking at the sodium-potassium pump. There's ATP. It's binding to one part. The pump moves. So they say conformational change in the hinge. And that change in shape is what lets the potassium ions be able to bind, and then they're released. Get another change in shape because of that. And you see the sodium ions are able to bind to the protein. And now we're transferring that phosphate group from ATP. Remember, it has 3 phosphate groups, so we're removing one. So we release now ADP, because there's only 2 phosphates, so adenosine diphosphate. And you get the release of those sodium ions. And then the protein goes right back to the original shape. Alright, and there you have it. That was the whole thing done out with this 3D model. Alright, let's look out, or not look out, but let's check out a breakdown of membrane transport. Bioflix, take it away.
We get all our energy and organic molecules from food. Before we can use the molecules we eat, they have to enter our cells, starting with the cells lining the small intestine. Let's zoom in to the surface of a cell. The plasma membrane is selectively permeable. Some molecules can move across it, while others cannot. How do materials enter and leave cells? Lipids, such as these yellow molecules, can dissolve in the lipid bilayer. Notice how they move down their concentration gradient, from where they are more concentrated to where they are less concentrated. This is an example of diffusion. Diffusion is a form of passive transport. It does not require energy from the cell. Most molecules can't cross the lipid bilayer. Here, the sugar fructose moves into intestinal cells by facilitated diffusion, moving down its concentration gradient through a transport protein. Facilitated diffusion doesn't require energy from the cell, so it's also a form of passive transport. Water crosses the plasma membrane by facilitated diffusion, or by diffusing across the lipid bilayer directly. The diffusion of water across a membrane is called osmosis. The sodium-potassium pump moves ions against their concentration gradient, from where they are less concentrated to where they are more concentrated. This requires energy from the cell and is known as active transport. Energy from ATP is used to move sodium ions out of the cell and potassium ions in. Another type of active transport is CoTransport. Here, both sodium ions and glucose move into the cell through a CoTransporter protein. Sodium ions move down the concentration gradient created by the sodium-potassium pump, and glucose moves against its concentration gradient. Now let's move to the other side of our intestinal cell. Materials can be exported in vesicles that fuse with the plasma membrane and release their contents outside the cell. This process is called exocytosis. In endocytosis, the plasma membrane pinches in, forming a vesicle that contains material from outside the cell. On this side of the cell, we can also see oxygen and carbon dioxide diffusing across the lipid bilayer. Cells use all these processes to get what we need.
Okay, this time she went further than I wanted her to, but whatever, it's good. You can always go back and rewatch that part. We haven't talked about the CoTransport thing yet, but I'm gonna lay down real quick talk about it. Alright, so back to the sodium potassium pump just for a second. So you're getting here kind of like the cycle that it follows. You have a picture 1, so picture 1 is right here, and then it moves to 2, 3, so on and so forth. So here's my plan. I'm gonna walk you through it step by step, and you just need to follow the movement of these ions. Alright, I'm not gonna do it on this slide. There's it's broken down in I think 3 different slides here, so you see 2 processes or 2 steps happening in each one. Alright, figure 1, they just described that there is some sodium in the cytoplasm. It's going to bind to the pump and it's going to help it to change its shape. Feel free to pause and read all of the words that are there. In the second step, you have the sodium binding that helps to bring forth this ATP. They call this thing, when the sodium binds, they say that it stimulates phosphorylation by ATP. In other words, the ATP is going to drop off a phosphate group. Remember, it has 3. It's going to leave 1 with the pump. That is the energy needed to make the pump move. So then after it's phosphorylated, you get the change in the pump. It opens out, outways. It spits the sodium ions out. The potassium ions can conveniently come and bind in. Alright, and then you had that one last movement where it's opens up and then it just drops the potassium. And then the cycle just keeps repeating. So now that we have done all the types of transport, let's just review really quickly in this picture. So purple molecules right here, that's diffusion. It's passive transport. There's no energy required. Then next to it, we have what we call facilitated diffusion. So now we're involving those membrane proteins, but again, there's still no energy required. So the one that's dead center is like a channel that's always open. There's a constant flow. And then the one that looks kind of like it's open mouth Pac-Man, going into the cytoplasm, it's dropping off those solutes that it picked up from the outside. Again, no energy needed. And then finally we have the one that we just covered, which is active transport. So this one, the concentration gradient is ignored essentially, and the pump is just going to move ions as necessary. Alright, so you might already know this, but I'm going to state the obvious anyway. So your body, it actually has its own electrical current, and this is why if you get hit with a bolt of energy, so if you get hit by lightning, or if you stick something metal into a socket, you get a shock. So why are you shocked? Because your body is conducting the current. That's why you're not supposed to keep electronic devices near the shower, or I don't know, there's just a lot of things you could do that could potentially electrocute you. So why is the current able to pass through your body? It is because of this membrane potential situation that we have going on. So the premise that I'm giving you is going to help us understand that first bullet. But basically, a membrane holds a charge, a positive or a negative charge. And if you know anything about electricity, current, even with magnetism, there is an element of having this positive in and a negative in, positive charge and a negative charge. Like, it keeps presenting itself over and over again. Your cell membrane is no different. If a charge builds up, you can essentially you conduct the flow or current through. So we call that membrane potential. So what causes the voltage is literally the variation in the anions and cations that are like on both sides, or they straddle the membrane. All right, so we have two forces that contribute to pushing the ions from inside the cell out, or vice versa, and those two are called together they're called the electrochemical gradient. So the chemical force and the electrical force combined, that's what we're looking at. The chemical part of it is dealing with the ion presence, and then the electrical force is what drives the movement, so hence, you know, electrochemical gradient. All right, almost done. The proteins that can create voltage, we call those electrogenic pumps. Sodium potassium pump is an example for animal cells, and things that belong to the plant kingdom or the fungi kingdom, and bacteria too. They have something else called a proton pump that's also at work. Can you guess what molecule it moves? Proton. Going back to chemistry again. So if you said hydrogen, ding ding, you got it right. And that's what they do. They're going to move hydrogen. So the whole point of these pumps is just to build up energy so that work can be done within the cell. What kind of work, Miss Johnson? The kind that involves chemistry. All right, here's your picture. Doesn't get any better than this. So you have, it shows you the charge on both sides of the membrane. That's the first thing that you want to look at. So negative on this side, positive on, oh, positive, come on, positive on that side. All right, and you know that these protons, they're carrying a positive charge. You literally are pushing them against their will. They do not want to go to the extracellular fluid. However, that proton pump and that ATP is going to make it happen. So we are going against the concentration gradient. The flow of the protons is not like just a natural thing. It needs active transport in order for it to occur. All right, we're almost done. I know I already said it once. This is the last time I'm going to say it. So there's some transport proteins. They require the help of another membrane protein in order for them to work. So this is that whole CoTransport situation that was covered in the BioFlix animation. So what happens is it's like a coupled reaction that has to take place. So you can't have one thing happen without the other. And essentially it's just like a cascade of events. So maybe something comes and binds to one pump and it inadvertently causes the other pump to respond. It's like a waterfall kind of thing. And we're going to look at an illustration of what they are talking about in bullet 2. So I'm going to go ahead to the next slide. Last slide. So CoTransport picture, boom, right there. You saw it in action in the BioFlix thing. Take a second, pause it, look at the image, figure out what's going on. So you have the proton pump moving the hydrogen ions across the membrane, and then you have a sucrose proton co-transporter. So in order for us to get those sucrose molecules or sugar carbohydrates into our extracellular or out of the extracellular matrix into our cytosol, we have to, not we, but plants and fungi or whatever else is going to use this. This is how they're going to make it work. So I'm all done. Good night.